Let's discuss about the detection of yeast and mold in food products such as dairy and bakery products. And as I earlier said, that microbial analysis is very much dependent upon food as well as on the microorganism and the strain, and they are complex methods. They may be different for different organisms and species, but we will be discussing some of the general approaches for determination of yeast and mold in food. So these methods are basically based on the enumeration approach of measuring the number of these pathogens within the food product as well as in the ingredients that are used for food processing. Another way of doing this kind of analysis, or the need for this analysis, can be we can scrape the surfaces from the processing area, because this yeast and mold can get into this processing environment from which it contaminates all the processed food. So this kind of analysis can be done on surfaces as well. If we talk about the principle of this microbial analysis, we need to have the nutrients that are needed by these microorganisms, especially yeast and mold, to grow in a specific media. And then the second thing is we have to add some of the antibiotics to these media, because antibiotics would be killing most of the bacteria and they would not show up in the final plates. Because if you don't add these antibiotics into these media, then the microbes, other bacteria can also grow along with yeast and mold, and it would be difficult to enumerate these yeast and mold in the food. So antibiotics is an essential component of these media. A normal process is it has to be incubated at a temperature of 22 to 25 degrees C for 3 to 5 days, because normally yeast and mold take a long time during this incubation period to show up their colonies, so they have to be incubated for longer time. And this method is a general purpose method, and it may not be suitable for determination of yeast and mold for specialized food. As I earlier said, that food might have higher salt concentration, higher sugar concentration, different water activities. So this general purpose method is not very much specific to every different kind of food. So if we look at the processes involved or the steps involved in this method, is we start with the preparation of media, then we prepare dilutions, and these are the cereal dilutions. Then we plate the agar or other media that is used, and then we go for incubation, as I said, at 22 to 25 degrees C for 3 to 5 days. And after that we count the colonies and look at the growth pattern of these colonies. These colonies are then differentiated according to their morphology. We have to see whether they are yeast or mold, or what kind of mold they are, and then we record our results. So these are the steps in the method of detection of yeast and mold. So if we, as I said, they are different for different kinds of food. So if we are dealing with some of the grains in which we want to measure the yeast and molds, then we use this media called as DG18, that would stand for Dichloran-glycerol 18. If we have to deal with these other yeast and mold in jams and fruit concentrates or other syrups with high sugar concentration, or dried fruit as well, then we have to add other media such as 20% sucrose that would be added to help the osmophilic fungi, osmophilic yeast or mold, because most of the osmophilic microorganisms are the one that can grow under low water activity or high osmotic pressure. So this would actually facilitate their growth. Another thing is added, that is malt extract at 50% sucrose concentration weight by weight. So these are the media for different kinds of food, as we earlier said. Then the molds have to be enumerated by a surface spread plate method. So the agar media has to be spread on a plate in a very thin films, so that these thin films would be not they would not be depriving these growing microorganisms of any oxygen or other atmospheric conditions, as well as the hot agar media can have some of the shock on these microorganisms, so they would not grow. So that is why this method of plating is very much useful. Now once we have incubated them in incubator for 3 to 5 days, then we normally count the number of colonies and distinguish between yeast and mold, looking at the morphology of the yeast and mold. Then what we do is we examine with the crystal violet strained smears that are used actually to differentiate between bacteria and yeast. Because bacteria might grow in the media, so these strains are actually used to measure, to see whether we are dealing with bacteria or yeast or mold. Food articles, again, for example, milk powders and meat or other particles can interfere with measurement of these colonies. So what we do here is we actually, in order to do the dilution, we do extra plates or extra tubes that are diluted to counter the effect of these particles, because these particles might interfere and our colony counting may be erroneous. So that's why we have to take extra steps, and extra tubes have to be processed in a similar fashion to avoid the effect that comes from the food particles, and so that we can have the proper amount of colonies counted. And we normally refrigerate the samples in a refrigerator for some time, and that actually helps to identify colonies versus the food particles. So this was the method for detection of yeast and mold in dairy products or bakery products.